All right. Happy Busch Gardens Day, everyone. How many of you are planning on going to Busch Gardens today at some point? How many of you are going dressed as a pirate? To scare the pirates. You know, it's pirate themed this year, right? What was the theme last year? Do you remember? Oh, you guys were freshmen. Last year was clowns. Yeah, right. This year's pirates, and I have to admit, when I heard pirates, I thought it was a little bit silly. That could go horribly bad. It could be like either cheesy or really good. So you'll have to see, right? Okay, so you guys have been out in the field the past 2 weeks, right? Yeah. Picking up deer scat, right? Counting trees, measuring stuff. All right, you got some data, right? Now what do you do? Okay, we're going to do stats. Woohoo! So I talked about it briefly last time. Okay? And I showed you, all right, everybody, simmer down. I know it's so exciting, it's Friday. I showed you one type of test. Okay? There are 3 that I want to show you. Okay? So what I'm going to do is I'm going to run through the first one again, and then I'm going to show you the other 2. And you want to be thinking about what you're going to do with this data. You want to be thinking about hypotheses that you can ask about this data. Okay? That's what you want to be thinking about, right? And these tests are going to be the ones that are going to help you to essentially look at those hypotheses and decide whether you can reject them or not. Okay? We good? All right. It's one of my favorite things on stats. Again, stats can be used very powerfully to get what you want in life, but not if you use it like that. Okay? That's going to make it look not so great. So be careful. With great power comes great responsibility. So, we're talking about hypothesis testing again, okay? So the example that I talked about last time is what's called a contingency test between 2 categorical variables. Okay, so whenever you have all this data, right, but you have different types of data, and you always have to ask yourself, what type is my data? What do I have, right? What am I looking at? So when you happen to have 2 different categorical variables, and you wanted to ask the question, are these variables contingent upon each other? Another way to say it is, are they independent of each other? Okay? That's what a contingency test can answer. Right? So test it as an association. All right? So here's the setup again. You've got this parasitism behavior in the host. You have trematodes, and they have 3 cycles. Okay, shorebird, and then this parasite, the bird poops in the water, the snail picks it up somehow. That's what an infected snail looks like. All that white in there is all parasite. Okay, the white that you're seeing here. And then the killfish eats the snail. And then, again, the awesome part is that this parasite goes into the fish's brain and wreaks havoc. And then somehow this fish gets eaten again by the bird, and the cycle continues. Okay. This is the life cycle of the parasite. And so I showed this video last time, and I asked, which one is infected, and it's like ridiculously obvious, right? This thing's going nuts. Okay, so this is almost like kind of a silly question, but we'll look at it. And in fact, whenever you do the test on this, you'll see that there is a very significant result, and I'll explain what significant means again in a second. So here's the experiment that was done. So you have 2 categories. Was the fish eaten, or was the fish not eaten? And then was it the fish infected? Was it lightly infected, uninfected, lightly infected, or highly infected? Okay. So level of infection is one categorical variable, and whether or not it was eaten is the other. And you want to ask the question, are they dependent on each other? Okay. So this is like the counts. This is a frequency table. They give you the counts. Here's the total number of fish that they actually ended up using in their experiment. Data visualization is very important. This is one way to visualize this kind of data is through what's called a mosaic plot. Okay? So you have a relative frequency here. Okay? And then you have on the one axis, you have the infection status. And on the other axis, the vertical axis, you have whether it was eaten or not eaten. Yellow means eaten, red means not eaten. And you look at that and you're like, well, yeah, the relative frequency of being eaten versus not eaten is definitely affected by whether you're infected or not. Right? So that looks to me like these guys are associated with these variables. Pretty clear. But remember, there could be uncertainty. This could just be random data. Just happen chance. You just happen to get data that look like this. Okay. So we need to use statistics to quantify that uncertainty. Is it random or not? Is there evidence for it being random or not? That's the idea. So there's our observed table up at the top. And then what you do is you create a null hypothesis. Okay? This is what hypothesis testing is all about. And a null is something that you really don't expect to be true. In this case, what do we expect to not be true? Well, we expect that, the null hypothesis would be that there is no association between the two variables. And given that there is no association, if you assume the null hypothesis is true, you get this type of table here. That's the expected table that you would get if there was no association between the variables. Notice that these column totals and the row totals and the total number of fish is the same in this table and the observed. Okay? So you're using this information to construct these numbers under the assumption of no association. Okay? So just look at it. If there were no association, you would expect to see this on average in your data. And look at what you have. It looks pretty different, right? But again, we want to quantify that difference. All right. So you create what's called a test statistic. Okay? And this essentially, what this test statistic is, has to do with this null hypothesis. Okay. This is a measure of your data relative to the null hypothesis. So what you're doing is you sum over the columns and the rows of your table, and in the numerator you subtract the observed and the expected, and you square it. So this is a distance between the distance from expected. Okay, how far is your data from what you would expect under the null? Okay. So that is giving you some measure of distance from your data to what you would expect under the null. If that is far enough or large enough, then you would expect that your data is evidence that it did not come from the null hypothesis. Okay. The hypothesis is false. That's the idea. If that number is big. But remember, what does big mean? It's a relative measurement. Okay, so you have to divide it by something. So in this case, you're dividing it by the expected. Okay, so it's sort of like a relative distance. Okay? Okay. So you get this number from your data, from those two things. And then you ask the question, how big does this statistic need to be to reject the hypothesis of no association? The larger it is, the more likely it is we would reject. That's the idea. It's further away from the null. Right, this is where that lovely p-value comes in. You guys have heard about this ad nauseum, I think, in lecture. Okay? From what I heard, you guys did pretty well on the exam with this stuff. But so the p-value, what is it? It's the probability of obtaining the data, or more extreme, assuming that the hypothesis of no association is true. Assuming the null. So this is, for those of you that know conditional probabilities, this is a conditional probability. Given that the null hypothesis is true, what's the probability of seeing the data or more that you actually observed? Okay? That makes sense? So it's a probability. And then usually, with hypothesis tests, we create this threshold value. Right? Because you have to have some measure of, well, how big is big? Right? How small does my p-value have to be? How unlikely is it, this probability, before I decide that the null can be rejected? So that's what alpha is. Probability threshold at which we would reject the hypothesis of no association when our p-value is smaller than this level. It's usually set to 0.05. And again, I think we talked about this in lecture. But remember, it's not really a threshold. If you got something that was 0.06, you might still think that there could have been there's some evidence of no association. So it's just standard practice to use 0.05. Okay, so related to this alpha is a critical value. This gives you a threshold value of this statistic, such that whenever what you compute is larger than that, you would reject the null hypothesis. Okay? And it's related to the significance level. Okay? Smaller significance, the larger that critical value becomes. So here we go. This is what you calculate from the data. That's the critical value at 5% significance, which is 5.99. this is much larger than that. So we would say that there's a significant association between being parasitized and then being eaten. And you can actually calculate it, thanks to computers. And you get your p-value of about 10 to the minus 15. Now again, that p-value is a probability. It says, if there is no association between the variables, I would expect to see that value or worse with this probability. Okay, think about that. 10 to the minus 15 is a probability. It's very, 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 very tiny. Okay. So yeah, I would say that you have pretty good evidence that there is an actual association between being parasitized and being eaten. And again, if you look at the picture, this fish is like breakdancing, right? It's pretty clear that it's going to be seen by a bird, right? It's going to be picked out. So we have evidence that parasitism does affect behavior. Okay? Remember, this is all about quantifying uncertainty and gaining evidence that you can never prove. Okay, so in the lab, pretty sure you'll be using a program out there that will compute this for you, do all this for you. You can put in your data, it'll do a contingency table, give you the results. This is the sample output. And you see all the values that we saw before. You see chi-squared value is 69.8, degrees of freedom is 2, and then the probability it says is 0.000. They didn't worry about the 15 decimal. Okay. Let's go on to the next. 2 sample t-tests. This is a different test altogether. Okay, here's the setup. You want to test whether the means of a numerical variable between 2 independent groups are significantly different. Okay. So what you have now is a categorical variable and a numerical quantitative variable. And you measure the same numerical variable in 2 different groups. And you want to ask, are the means going to be different? Okay. Now, I want to point out, this is somewhat of a silly question. Okay. How often do you think, if you have 2 groups, each group has the same kind of measurement on it, how often do you think you're going to get the exact same mean from these 2 groups? What do you think? Not very often. Okay. Almost probably 0 that would actually happen in a population. Okay. So it's kind of weird to be asking, is there a difference in the mean? Of course there is. Okay. But, so whenever you do these tests, you have to keep in mind that it's not just whether or not you reject a null hypothesis. In this case, that the mean would be the same. But you would want to know what the magnitude of that difference is. And even that magnitude, you can only interpret based on context and scientific thinking. Okay. So this is not just about tossing and proving something is wrong or disproving a hypothesis test. It's that you care about the difference in the means as well, how far apart they are. So just keep that in mind. Okay. Okay. So here's the setup.
And over here we have a very interesting situation. We have a yucca that has been utilized by a loggerhead shrike. These horned lizards were impaled on the yucca by a loggerhead shrike to either be eaten at that time or at a later date. And these are round-tailed horned lizards, one of the 3 species of horned lizards we have in Texas.
Good stuff. Yummy. Okay, so this is pretty cool. So this bird essentially just grabs a lizard and impales it on a yucca plant and says to himself, you know what, I'm going to save you for later. I'll come right back. Don't go anywhere. Right? Okay. And so one of the things that the biologists were wondering is if there's 2 groups, was the lizard killed or not? So that's your 2 groups. And they were wondering if the horn length on these lizards was different, the mean horn length between those 2 groups. Thinking being that those lizards that have long horns will probably be found more likely in the not killed group. Right? So somehow those horns are protecting it from predation. Yeah, that's the idea. So here's their data. Living group, killed group. They have a sample mean. So this is in millimeters of their horn lengths. Okay. And I mean, you can look at this and see that, oh yeah, look, this is larger than that. Right? So you would say, oh yeah, so maybe it's true that those horns are somehow protecting them from predation. But again, we have to quantify that. We want to say, is that random sampling? Is that just randomness? What's going on? Because remember, this is only a sample from the population. So you can compute the standard deviations, which is the measure of spread in each population, and then you have sample sizes there, which are pretty different. Okay. Here's a frequency histogram or distribution of the data. Living lizards up top, killed lizards below. Okay. And then again, horn length is the horizontal axis. And again, you can kind of see that the distribution is a little shifted more to the right for living lizards. Okay? So let's test this. If the means are significantly different. All right, what do? We create a test statistic, as before. Before it was called the chi-square. This is called the t-statistic. Right? So in this case, the numerator, and again, it's the same thing. It's a fraction. The numerator is the difference between the means. Okay? So again, that's measuring how far from the null. Remember, the null hypothesis is that there is no difference between the means of the two groups. Okay, I'll say it again. The null hypothesis is that there is no difference between the means of the two groups. And so that numerator kind of captures that distance from the null. Right? The larger that distance is, subtract that number is, the further it is away from that null hypothesis. But again, you have to do it relative to something. Okay? So you might have large, but it's got to be large relative to the natural spread of the data, or standard deviations of the variation. Okay? So this is a nasty formula, but don't worry about it. You have two different groups. Therefore, you have two different standard deviations and two different variances. That's what the squared is. So you have to take essentially a weighted average of those two. Okay. N1 is the sample size of group 1, N2 is the sample size of group 2. So you're just doing a weighted average to create what's called a pooled variance. And then down here, in the denominator, is the measure of spread in your data. So you're normalizing that difference by the natural random uncertainty that you have. The larger you have natural spread in the data, the larger that difference between the means has to be before you would say that there's a significant difference. Okay. So again, how big does this statistic need to be to reject the hypothesis of no difference in means? Okay, here we go. Here's your t-statistic. We calculated it at 4.35. Here's your critical value, 5% significant, 1.97. That is larger than that, and therefore you would say that there is a significant difference. Okay. So again, the word significant does not mean important. Okay. It doesn't even mean that the difference is really large. It just means that it is a statistically significant difference. You've rejected a null. That's all it means. Okay. P-value. I'll say it again. If there is no difference between the means of these two groups, you would expect the data that you got, or worse, with this probability. That's pretty small. Right? It's a good deal smaller than 0.5. Okay? Does that make sense? You guys bored out of your mind? You want to go be chased by pirates? Is that what you're saying? Yes. Okay, so I only got one more and then we're done. Evidence that long spikes do protect horned lizards. Okay, that's what we have. So again, you can use the computer to do this test for you. This is what a sample output looks like. I subsampled the output because this is like 130 something points. I didn't want to put it in point by point. You got a t of 0.5, 0.05. Here's your standard deviation. That's the pooled one. And here, again, it's so small that it just reports 0. It's not actually 0. Okay. Okay. Last one. Well, it's actually 2, but it's all on together. Now you have 2 different numerical continuous variables. Okay. It's a lot like the contingency test, except now you're doing it for 2 continuous quantitative variables. So you want to test if there is an association between 2 numerical variables, which is the correlation. And if so, you might want to model the relationship between them. And that clearly would be regression. Okay. This goes back to that lovely phrase, correlation does not imply causation. I'm sure you've all heard that. And so regression you tend to think of as a causative thing, whereas correlation is the other way. Okay. So I'll explain this in a second. Okay, so what is the correlation coefficient? Measures the strength and direction of the linear association between two numerical variables. So you have a variable x, you've got a variable y, you can plot them in a scatter plot. Okay, these are all your data points. And then what you do is you take that box in the middle, that's the coordinate corresponding to the mean of your x variable and the mean of your y variable. That goes right there. Okay? Create a quadrant, center of that point. And then what you say is that where are all my points relative to that middle square? Okay? If they're all in the positive deviation in y and positive deviation in x, and mostly in the negative deviation in x and negative deviation in y, then you would get a positive slope linear relationship between those points, between those variables. Okay. So our R, again, this is our sample estimate. For our population, we don't have access to all the data, but for a sample, we do. You take the distance between x and its mean, so that would be this distance between that and here, times the distance between y and its mean. Okay, so this numerator is going to be positive whenever you're over here or you're over here. Because if you're to the left of x and you're to the left of y, you're in this quadrant. So this is going to be a positive when you're here or here. And it's going to be negative when you're here or here, which corresponds to a negative slope. Okay. And again, you have to normalize. So this denominator is just a normalization factor. So that R is between -1 and 1. Okay. But the most important understanding is the numerator. Okay. All right, there's some examples. So R is 0 on the left. R is about 0.5. So it's positive, right? So the sign tells you what direction that relationship is. And then the size of it, if it's closer to 1, tells you something about the slope. Not actually doesn't tell you about the slope. It tells you how closely they are together. So for example here, 0.5, you have somewhat of a positive correlation. That's a little bit of a larger negative correlation. And here, it's 0.9, it's very close to 1. It's a very strong, linear, positive correlation. Okay. All right, I love this example. Okay. Mainly digits. So we got two different variables. There's these CAG repeats, CAG, they're found in the androgen receptor gene. And the number of CAG repeats is known to correlate positively with sensitivity to testosterone. So all the guys are going like this now. So you take your right hand, okay, and what you do is you're relating that number of CAG repeats to this 2d40 ratio. Okay. So they've shown in other studies that the ratio between the length of your second digit with the length of your fourth digit correlates with testosterone. Okay. So I think it's sort of like if it's larger than 1, you have more testosterone. If it's less than 1, you have less testosterone. Don't mess with your names. So yeah, so my 2d40 ratio is larger than 1, so that would mean I have a lot of testosterone. And my balding might give you another indicator of that. Okay. Okay, so here's the scatter plot. Number of CAG repeats, 2d40, and all of them are like, what does that mean? Okay, so here's the question. Do the number of CAG repeats also correlate with this 2d40 ratio? Kind of looks like it does, right? Well, let's see. We get an R of 0.3082. But is that significantly different from 0? Okay. No correlation is R equals 0. Anything non-zero would be called, you would say they were correlated. But, this was only for a subsample of all the population of males out there. And so the question is that just from random sampling, 0.3, or is that really a good estimate for the population you see? Okay. So we're going to create our test. Our null hypothesis is that there is zero correlation. Okay. So the null is there is no correlation, no association. So what we do is we create this test statistic. It's just R in the numerator, the correlation estimate, divided by what's called the standard error of R, which looks like that. Okay? Don't worry about it. Formula. Use it. Okay. And again, it's a fraction. So how far are you from 0? Numerator. How far are you from the null? Divided by some sort of correction factor. Okay. Here's our calculated t. And there's our critical value. They're really quite close. So you would still say that there is a statistically significant correlation between them. In other words, you have evidence that it's a non-zero correlation. Okay. P-value is 0.0372. Not that far from 0.05. So you want to take that number into account. Okay, where it is relative to 0.05 matters. Okay, in particular based on past evidence. Okay. But still, you would say that there's evidence that the number of CAG repeats does correlate with 2D40 ratio. Okay. Yeah.
If you were to publish that, is there, like if you said weak or strong, is there a quantifiable way to say what people know?
It varies a lot. People will put lots of adjectives on it.
Yeah.
Like there's a strong correlation, strong significance, weak significance. Or if it's just over 0.05, we'll say barely not significant, things like that. But all these words don't have
Okay. Significant is the only one that actually is attached to
it's just,
real.
yeah, and it's just straight up based on that threshold. Yeah. Okay, a couple of things. Correlation depends on range. Okay, so depending upon what your measurements are for log of body mass, for example, at the top, if you got measurements all the way from -4 to 4, you would say that there's looks like a pretty strong negative correlation. But if you did a sub-region, all of a sudden it doesn't have as much of a strong correlation. So it's very important to keep that in mind. Also, measurement error can screw things up, right? So maybe in the population, you have a very, very, very strong positive correlation on the left. But as you have increasing measurement error, what's going to happen is that cloud of points is going to spread. And it's going to weaken your correlation. Okay? So measurement errors are very important thing to keep in mind with this stuff. But you could do replication to reduce that. Okay? So the more replicates you do for your experiment, the better you can correct for that type of measurement error. All right, this is a well-known XKCD comic. I used to think correlation implied causation, then I took a stat class, now I don't. Sounds like the class helped. Well, maybe. Good stuff. Another great one. This is from the Pastafarians. They are part of the Flying Spaghetti Monster group, where they worship the Flying Spaghetti Monster. They're atheists. And this is one of their lovely things. It is known that the number of pirates correlates positively with the global average temperature. Right? Therefore, if somehow, why should we fight climate change? If we're able to somehow reduce the global climate temperature, what would happen? Notice this is going the wrong way. This is lower pirates than higher pirates. So it's more like a negative correlation. Yeah, you get more pirates. So we should not try to help the world from warming, because otherwise pirates will come back. So yeah, that's a great example of correlation does not imply causation from the Pastafarians themselves. Okay, last thing for you guys is regression. Okay, it's the sibling of correlation. And you use it when you're trying to talk about a causative effect, essentially. You're trying to, because in this case, you're going to have an independent variable and a dependent variable. So by definition, if you have something called a dependent variable, it is is dependent on the other one, and therefore a causative relationship is implied. Okay. So the idea is you want to do prediction. This is whenever you use this stuff. There's a lot. When you want to predict something, you use regression. So you predict one numerical variable from that of another. It uses linear relationships if you use linear regression. And of course, there's nonlinear regression that you can do as well. But we'll talk about linear regression. What's the formula? It's a line. Okay. A is your intercept, B is your slope. Nothing too exciting. Algebra 101. And your slope is going to be positive here, negative here, 0 here. And your A, which is your intercept, controls the vertical up and down of your line. Okay? Displacement. Okay, so here's an example using the method of least squares. So this is one way to do linear regression. Okay? It's an optimization procedure. You're trying to find the best fit line to your data. So in this example, you have black pigmentation on the noses of male lions as a measure of age. Okay? So you've got two quantitative numericals. One is the age of the lions, one is amount of black pigmentation. Okay? So as you can see here, the x-axis is proportion black, and the y-axis is age in years. And what you're seeing is three different types of possible lines to those fit that data. Now the idea is that when you put a line down, think like pickup sticks. Right? Put a line down, you measure the distance between the line vertically and all the points. And you think of it as an actual distance. You sum it all, and you sum it all up. And you're going to get a number. Pick the line that minimizes those deviations. Okay. So you have large deviations here, smaller deviations here, and then this line would give you the smallest deviations over all possible lines. Okay. And that would be your linear regression. That would be your optimized line, best fit. Okay. Now, don't ask me, I mean, you could do this with any data. It could be the most disgustingly spread out cloud of data ever. You can always do this. You'll get a best fit line. And then you have to say, well, so what? Okay. So you always have to ask, is that a good fit? Right? I mean, is that like, would I say that this variable really explains that one? Right? Okay. So you minimize sum of squared deviations. Okay? That's what you did. You can write down what the slope is, you can write down what the intercept is in terms of the means. So notice that B and A are written solely in terms of the data and their means. Okay, so you find the slope first, and then you can compute the intercept from that. That looks a lot like the regression coefficient, the correlation coefficient. It just so happens that you can write it as R times the ratio of the standard deviation. Fun stuff. All right, there's your line. Okay. How big is your slope? Pretty small. So notice that if R is 0, B is 0. Right? If R is not 0, B is not 0. Okay, so sample output. Throw it in the computer. You can get 47 data point pairs. You'll get your A, you'll get your B. 5 minus 3. You get your R, so it gives you your correlation, and it gives you a p-value. Okay, so that p-value is telling you whether or not your R is significantly different from 0. So it's automatically testing the hypothesis of is there a correlation in these variables or not. Okay. So in this case, it's less than 0.05, so you would reject the null hypothesis, which states that there is no correlation. Okay. So you have evidence that there is correlation. Fun stuff. There it is. It even gives you a graph. How nice. Okay. Okay. Now, here's the final slide for you guys. Deer, ticks, and trees, right? That's what you guys did, right? Okay, so deer, poop, ticks, and trees. Some questions to think about. Are deer lowering the carrying capacity of the college woods? In particular, are they altering the availability of nut mass, the most valuable food in forests? Does forest composition, particularly presence or number of oaks, affect tick abundance? And is there a relationship between abundance of deer and abundance of ticks? A lot of these questions we're going to ask can I answer them or have prepared a hypothesis using the data that I've collected to give me evidence for any of these types of questions? For a hypothesis for these kind of questions. Okay. So you just want to, before next week, you want to be thinking about the data that you have, thinking about these types of questions to construct your own hypotheses, and then you're going to do your own data analysis. Good? I can definitely quiet today. Any questions? Oh, the attendance sheet. Oh, you guys. This is Monday's discussion.
Okay, should I make?
Does anybody have an attendance sheet for that?
I'll make one.
Oh, man. Yeah, I know. No, everyone should sign. Yeah, make multiple. Someone tear a piece of paper for your row, whatever. Make sure you get your name on a piece of paper and have it up to you.